Welcome to this lecture on the structure of proteins. Today, we will explore the 4 levels of organization that give proteins their unique biological functions. Proteins are complex macromolecules essential for life. Let's begin by defining what makes a peptide a protein. By definition, a protein is a relatively long peptide chain with at least 40 amino acid residues. Shorter chains are simply called peptides. Understanding protein structure requires examining 4 distinct levels of organization, ranging from simple sequences to complex folded shapes. Primary structure refers to the unique, linear sequence of amino acids linked together by peptide bonds. This specific order is not arbitrary. It is the fundamental blueprint for the protein's eventual 3-dimensional shape. The peptide bond itself is rigid and planar, forcing the protein backbone into a zigzag arrangement of atoms. Moving beyond the linear sequence, we look at secondary structure, which describes the local spatial arrangement of the backbone. The alpha helix resembles a coiled spring, held together by hydrogen bonds between NH and C equals O groups along the backbone. Alternatively, the beta-pleated sheet consists of extended protein segments held together in a parallel or anti-parallel orientation. Hydrogen bonding between the carbonyl oxygen and the amide hydrogen is the key interaction that stabilizes these secondary structures. Tertiary structure defines the overall 3-dimensional shape of a single peptide chain. This folding is driven by interactions between the amino acid side chains, or R groups, that are far apart in the primary sequence. These interactions include covalent disulfide bonds, electrostatic salt bridges, and hydrophobic attractions between nonpolar side chains. Quaternary structure is the highest level, found in proteins made of multiple independent peptide chains or subunits. These multimeric proteins organize subunits using the same noncovalent interactions that define tertiary structure. Hemoglobin is a classic example, a tetramer composed of 4 subunits working together to transport oxygen in the blood. Finally, we must discuss denaturation, the disruption of a protein's 3-dimensional shape, and the resulting loss of function. Factors like heat, changes in pH, or chemical agents can break the stabilizing bonds, causing the protein to unfold. Whether denaturation is reversible or irreversible depends on the severity of the damage to the protein's native structure. In conclusion, proteins are remarkable molecules whose biological function is intrinsically linked to their complex multi-level structural architecture. We have reviewed the 4 levels of organization: primary sequence, secondary backbone motifs, tertiary 3D folding, and quaternary subunit assembly. Maintaining this structural integrity is critical for all biological processes. Thank you for joining this overview of protein structure.